Good morning. Welcome to Life Science. The last time I saw you, we started the monohybrid genetic crosses. We did one or two examples. I would just like to revise that a little bit by going over a few more examples or activities in your book. We're going to start with number 3, activity 5.3, page 74. So if you could please get your textbooks out and your books out. We have done number 2 together in class. We're now going to do number 3. Okay, so number 3 reads, 2 hybrid tall plants are crossed with each other. Use a genetic cross to show the possible genotypes and phenotypes of the offspring. Also indicate the genotypic and phenotypic ratios. Right, so our first clue there is the word hybrid. Another word for hybrid is heterozygous. Now, if we remember heterozygous, the 2 alleles are different. We have one dominant allele and one recessive allele. Okay, so now if we remember how to lay out a genetic cross, let's number this number 3. We first have a key. Okay, you guys can do this in your book as I am doing it, so we are doing it together. Okay, the key, we're looking again at the height in plants. Okay, tall and short. It's 2 tall plants. So our key, if it is heterozygous or hybrid, okay, it means we will have the alleles being different. One capital T for tall and then one small t for short. So let's put that in our key first. Right, capital T. Okay, tall is dominant because we've got heterozygous plants that are tall. So that means the dominant allele has masked the recessive allele. Okay, and that's why the plants appear tall in heterozygous plants. Okay, then we lay out our genetic cross. If you remember, we had a silly little rhyme. It was Pip got me greedy for fig pie. Okay, and that stands for, say it with me, phenotype, genotype, meiosis, gametes, fusion or fertilization, genotype, phenotype. It tells us that 2 hybrid plants are crossed with each other. Okay, so first we've got to the phenotype. We are stating in words the 2 types of plants that are crossed. So it is tall crossed with tall. The genotype, the alleles, we said they were both heterozygous. So it'll be capital T times, capital T with a small t times capital T with a small t. Now remember meiosis, we leave open, okay, The gametes, remember through meiosis, okay, our two alleles will get split. So we write T, comma T times capital T, comma T. Okay, we now have 4 separate gametes. They will fuse, okay, in fertilization, and we then need to do a Punnett square. To show the crossing. Okay, we will take this first one and put it on the top here. And the second one, we will put it at the bottom. And then we cross. Okay, this first square will be 2 capital T's. This square will be that capital T with that small t. The capital letters always come first. Okay, this square will be capital letter first, small letter. This square will be these two together. It doesn't matter which one comes first, they are both the same. Okay, fusion we leave open. Genotype, we're now going to write down the results over here. So we've got 1 capital T, 2, okay, heterozygous. This is homozygous, remember, homozygous, dominant. Okay, then we've got 2 heterozygous. And we've got 1 homozygous recessive. Okay, that's our genotype. Now we need to describe this in words in their appearance, whether they are short or tall. So this one is homozygous tall. That's 1. This also, even though it is heterozygous, okay, tall, the tall allele is dominant, so phenotypically these two plants will appear tall. So we've now got 3 tall. This one is homozygous recessive. The small t stands for short, so we have 1 short plant. So in words, 3 tall, 1 short. Right. Okay, now we move on to number 4. A butterfly homozygous for brown wing color is crossed with a butterfly homozygous for pink wing color. All the F1 butterflies had brown wings. Okay, so if you remember, F1 is the first filial generation, that is the offspring. Okay, now 4.1 says, which is dominant, brown or pink wing color? We have to look at that last sentence. All the F1 butterflies had brown wings. That must mean that brown is dominant over the recessive pink wing color. If all the offspring have brown wings. Okay, so 4.1. We write brown wings. 4.2. Give a reason for your answer in question 4.1. As we already discussed, all the offspring of the first filial generation had brown wings. This tells us that brown is dominant. Okay, 4.3, we now have to represent a genetic cross to show how you will arrive at the offspring with brown wings in the first filial generation. Okay, so first things first, we have to have a key. We know now that brown is the dominant allele, so we will have a capital B for brown winged. Okay, therefore pink, we will still use the same letter. We will not use a P, we will use the B for brown. But because it is recessive, we will use a small b for pink winged. Okay, then again, we have our silly little rhyme. Pip gets me greedy for fig pie. Phenotype fusion or fertilization Okay, so we are told that we are crossing a brown homozygous with a pink homozygous butterfly. So in words, we will say brown winged times or cross with a pink winged. Right, now we've got to write the alleles for the genotype. We were told that they are both homozygous, so the alleles will both be the same. In order for us to have a homozygous brown winged, okay, they must both be capital B. Pink winged, they have to both be small b's. Meiosis, we leave that line open. The gametes, this has been split through meiosis, so we write it like this. Fusion, we leave open, and we will then do the Punnett square to show how these gametes have fused. We'll take this one first, put it at the top, this one second, put it at the bottom. Okay, this first block, capital letter always first, capital B, small b, capital B, small b. And you'll see that's the same for all of the blocks. They're all capital B, small b. Right, now we will fill that in, the result we will fill in at genotype. There are 4 capital B, small b. In words, that means we have 4 brown-winged butterflies, because the dominant brown allele masks the recessive pink allele, and so they will all appear brown-winged. Right, 4.4. If the first filial butterflies, or the offspring that we've just seen, were allowed to interbreed, use a genetic cross to show the genotypes and phenotypes of the second filial generation. In other words, the offspring of the first filial generation. Okay, we always have our key first, even if we've done it in the previous question. Again, it's the same thing. Okay, but now it is not the P1 generation. Okay, it's now, we're taking the F1 generation, we're interbreeding them. They now become the P2 generation. Okay, so it's not P1P anymore, it's P2P. The rest is more or less the same. We've still got Geds, me, greedy, 4. Now fig, it's not F1G, it's F2G. Okay. Right. And we fill in the rest. Okay, so we saw that the offspring of the previous group were heterozygous brown-winged. Okay, we are going to breed 2 of these with each other. Okay, so it will be brown-winged crossed with brown-winged. They are heterozygous. Meiosis we leave open. Our gametes are split with a comma. Fusion we leave open. And we do another Punnett square. Cross those 2 together. Those 2 together. Those 2 together. The capital letter comes first. And then those 2 together. We will write our results over here by genotype. We have 1 double big B, 2 big B small b, and 1 double small b, homozygous recessive. Okay, our description in words. This is 1 homozygous dominant, so it's definitely 1 brown. This is 2 heterozygous, but the brown is dominant, so that's 2 brown again. And then the last one is 1 pink-winged. So we have 3 brown-winged and 1 pink-winged. last question we're going to do from this activity. It asks us to state the genotypic and phenotypic ratio of the F2 generation. So we just take that there, these semicolons can become colons. And phenotypic. Again, we're just changing the semicolon to a colon so that it is a ratio. Okay, and it's as simple as that. Right, so that is activity 5.3, number 3 and number 4. We are not going to do number 5 or number 6.